In this pencast, we're going to take a look at the structure of solids, specifically three of the cubic crystal lattice structures, the simple cubic, the body-centered cubic, and the face-centered cubic structures. We're going to start by just reviewing some of the characteristics of these structures, and then we're going to take a look at some of the problems that were given out in class, where you're asked to calculate things like density and radius and length of sides of cells. So just to jump in, we're going to begin with the simplest of the cubic crystal lattices, the simple cubic structure. And let's just point out some things that we should know. The simple cubic structure begins with a square arrangement of atoms in the first layer, and they touch each other along each edge, up, down, and sideways. So basically, it's called a square arrangement because if you connect four of the centers like this, you see a square pattern. It's very important to note that the atoms touch each other along the edge of that square. In the second layer of the simple cubic structure, atoms are placed directly on top of the atoms in layer 1. In the third layer, they're placed on top of the atoms in layer 2. Which leads to an algebraic description, A, which implies that each layer of the units of the crystal lattice is identical. Okay, each layer is found directly above the atoms in the layer below. When you look at the edges of the cell, you notice that two of the atoms always touch along an edge, which leads to the statement that two radii must equal S, where R is the radius of the atom and S is the length of the edge of the unit cell. So 2R equals S in a simple cubic structure. Finally, if you think about how the atoms are laid out in just one unit cell, you know you have a cube with 8 atoms, one at each of the 8 corners of that cube. And so since 1/8 of an atom at a corner is inside the unit cell, then those 8 atoms multiplied by 1/8 of an atom per corner gives us 1 atom per cell. Okay, so there's actually 1 atom contained within each unit cell of a simple cubic lattice. We calculated the packing efficiency of simple cubic, and we found that it was a 52% packing efficiency. And finally, the coordination number for each atom in the cell, the coordination number is the number of closest neighbors. If you pick one of those corner atoms, it's touching an atom below it, above it, to the left, to the right, in front, and in back. So it's actually got a coordination number equal to 6. Each atom touches 6 other atoms, one above, one below, one behind, one in front, one to the left, and one to the right. So the coordination number is 6. In a body-centered cubic structure, we'll abbreviate these, by the way, SC for simple cubic, BCC for body-centered cubic, and FCC for face-centered cubic. In the body-centered cubic structure, you still begin with a square pattern of atoms in layer 1, but when you go place the atoms in layer 2, you smarten up and you place layer 2's atoms in the holes that were in layer 1. So layer 2, you get an atom in the hole there, an atom in the hole, etc., like that. Okay? Layer 3 would have atoms in the holes of layer 2. With a bit of imagination, you can see that the atoms in that third layer will end up directly above atoms in the first layer. Atoms in the fourth layer will be above the atoms in the second layer, and that continues throughout the crystal lattice. So algebraically, we describe it as A, B, A. In my drawing, I was a little bit sloppy because I showed here the atoms still touching along their edges. When you place the second layer of atoms in the holes that were in layer 1, it forces the atoms apart. So a more accurate picture of layer 1 would have been atoms like this, still forming a square pattern. If you connect 4 of the centers, 1, 2, 3, 4, you still get that square pattern, but we notice they no longer touch along one edge. So 2R is not equal to S. When you place the center atom, the atoms in layer 2, though, on top of layer 1, we notice that the body-centered atom, the atom that will end up in the center of the unit cell, it rests on top of these 4. So it actually is touching all 4 of those atoms. Then when you put the third layer on, 4 atoms on the top, the 4 atoms that form the 4 corners of the top of your cube, will rest on top of that center atom. That means the center atom is actually touching 8 atoms around it, and so its coordination number is actually going to be an 8. When you envision that, you can see that 3 atoms are touching each other in a row along the diagonal of the entire cube. If you envision one atom at the corner and the bottom, it's touching the center atom, the body-centered atom, which is touching the top corner opposite that. So that means if we picture 3 atoms touching in a row like that, and I just connect their centers, you can see that we're going to get 4 radii, the radius, 1 from this atom, 1 from this atom, and 2 from the center atom. 4 radii form the diagonal of the cube. Now the diagonal of a cube, we showed with a bit of geometry, is equal to the root of 3 times S. So for a body-centered cubic structure, 4 radii is equal to root of 3 times S. How many atoms are inside this cell? Well, we still have 8 corner atoms, so we still have 8 times 1/8 each. So we get 1 atom at the corners. But we also have the 1 atom in the center, the body center. So when you add the 1 atom from the corners to the 1 atom body center, you get 2 atoms per cell. That's very important, along with this formula, 4R equals root 3S. We calculated the packing efficiency for this, and we saw that it was increased. If I recall, it was 68% packing efficiency. And I'm running out of room here, but the coordination numbers, we already talked about the body-centered atom. The body-centered atom had a coordination number of 8, and I'm going to leave it to you to see if you can reason for yourself that one of those corner atoms also has a coordination number of 8. Okay? You think about that. Each corner atom is touching 8 other atoms. I'll let you figure out how to rationalize that. So whether you're the body-centered atom or a corner atom in the BCC structure, your coordination number is 8. The face-centered cubic structure is the most complicated one in terms of visualizing it. You start not with the square pattern that we saw earlier, but you start now with what we call a hexagonal pattern. Notice that when you're filling in the rows in layer 1, you no longer put them in a square pattern. Instead, if you connect these atom centers, you get a hexagon, so we call that a hexagonal arrangement. In the PowerPoint presentation, we saw that when you put layer 2 on top of layer 1, it goes into the holes of layer 1, no problem. When you've done that, though, if you recall, there are now 2 different types of holes in the third layer. Holes that were above holes in the first layer, and holes that were above atoms in the first layer. Depending on where you go and put the third layer, which of those holes you choose, you get 2 different structures. One would be leading to a non-cubic structure. the hexagonal closest packing. That would be the ABA arrangement again. The other one was when we put the atoms in layer 2 in layer 3 in the holes that were above holes in layer 1. So that led to an arrangement called ABC. All right, and then it starts repeating. So ABC, ABC. I'm not going to be able to explain it in as much detail here, but we showed that 3 atoms, if you looked at one face of the face-centered cubic structure, so one face of the cube, this is what you'd end up seeing. You'd see 4 atoms at the 4 corners, and then in the center of the face of each square, of each face of the cube, there's an atom sitting there. So this time we have 3 atoms touching each other, so 4 radii again. But this time, instead of it being across the diagonal of the cube, it's across the diagonal of the square face of the cell. And we remember some geometry, the diagonal of a square face is the root of 2 times s. So 4r equals root of 2s. If you looked at any face in a face-centered cubic structure, this is what you'd see. So it's called face-centered because each of the 6 faces of the cube has an atom sitting right in the center of the face. Again, the atoms along the edges are forced apart, just like in BCC structure, so they no longer touch like that. But 3 atoms touch along the diagonal of the square face. How many atoms are in the cell? Well, we have our 8 corner atoms, so 8 times 1/8 will give us 1 atom, and that's at the 8 corners. Then we have 6 face-centered atoms. Each of the 6 faces has an atom sitting in the center. That atom sitting in the center is being shared equally by 2 cells, the cell that we're in and its neighbor. So half of each face-centered atom is in the cell, which would lead to 3 atoms from the face centers. Adding that together, there are 4 atoms per cell. When we calculate the packing efficiency, and we actually haven't done that yet in class, but if I recall, it's about a 76% packing efficiency. It might actually be 74. It's in the mid-70s like that. Okay, so 76% packing efficiency. Okay, I might be off by just a little bit there. But the point is that this is the closest packing because it's got the highest packing efficiency. So face-centered cubic is sometimes called cubic closest packed instead. It means the same thing. So sometimes you'll see FCC, face-centered cubic. Sometimes you'll hear cubic closest packed, and you have to realize that means the same thing. The coordination numbers I don't think we've discussed yet, so I'll leave that for you to discuss in class. So that's some of the background information that we've got. And you notice in terms of doing problem solving, there are 2 things that are important for each type of cell. You need to know the algebraic relationships between the radius of the atoms and the sides of the cell, the edges of the cell. You also need to know the number of atoms per cell. So 1 atom per cell, 2 atoms per cell, and 4 atoms per cell. All right, so now I'm going to jump in and do some of the problems that were on the sheet that were given out in class. So this is question 1084 from the handout, 10.84. The question reads, copper crystallizes in a face-centered cubic structure with an edge length, each unit cell's edge length, of 362 picometers. And we'll describe that in just a moment. It tells us to calculate the radius of each copper atom in picometers, and then to calculate the density of the copper in grams per cubic centimeter. So let's start. We know that it's a face-centered cubic structure, and we know we have copper. So I'm going to just start organizing some of the information we know. We know it's copper. We know that it's a face-centered cubic structure. Looking above, that tells me two important things. It tells me that 4 radii must equal root 2 times s, where r is the radius of the atom and s is the length of one edge of the cell. And it also tells us that there's 4 atoms per cell. Now we also know that the length of one edge of the cell, s, is equal to 362 picometers. And this may not seem obvious, but from a periodic table, we can look up the molar mass of copper. We're going to need that when we go find density, so I'm going to put that here. Its molar mass is 63.55 grams per mole. So let's jump in and start calculating. The first thing we were asked is, what's the radius of the atom? Well, that should be straightforward. We have this formula, 4r equals root 2s, and we know what s is. So let's start by finding the radius. So radius will equal root of 2 times s divided by 4 equals root of 2 times 362 picometers divided by 4. A picometer is a very small distance. Okay, you know some common SI prefixes. You know that a millimeter means 10 to the minus 3 meters, while some other units that are not so familiar, a micrometer means 10 to the minus 6. Let me just write these on the side here. So a millimeter would be 10 to the minus 3 meters. A micrometer, which kind of looks like the letter U but has a leading tail like that, is 10 to the minus 6 meters. A nanometer is 10 to the minus 9 meters. You can see a pattern. We're just going down by 1,000th each time. And that leads us to the picometer, which is 10 to the power of minus 12 meters. So a picometer is a very small distance. So the radius of this atom, grabbing your calculator, pause here and do the calculation, please. So square root of 2 times 362 divided by 4 gives me a radius following the sig figs here. Well, we've got 3 significant digits in the radius, in the length of the edge of the cell. So I'll write 128 picometers. There's the length of, the radius of the atom. The second thing we wanted to know was the density of the copper. Well, we know from grade 9 science that density is a mass divided by a volume. Well, mass of what and volume of what? In this context, the mass will be the mass of the cell and the volume of the cell. So of one unit cell. So let's stop and think about that. The mass of the cell. Well, the cell is made up of 4 atoms. We remember that for face-centered cubic structure. 4 atoms of copper make up the mass of this cell. Right? That's the only thing that makes up the cell. So we can calculate the mass of a single atom of copper. We know that its molar mass is 63.55. So if we take 63.55 grams, that's the mass of a mole of copper. If we divide that by Avogadro's number, that'll give us the mass of one atom. Okay, you remember there's Avogadro's number of atoms in 1 mole. Then the mass of the cell would be 4 times that, because there's 4 atoms per cell. Now notice, stand back and look at the bigger picture. If you take the molar mass of an element and you divide by Avogadro's number, you now know the mass of one atom. If you then multiply by 4, because we had a face-centered cubic structure where there's 4 atoms per cell, then we know the mass of the cell. If this were a body-centered structure, we'd be multiplying by 2, where there were 2 atoms per cell. In a simple cubic structure, we wouldn't multiply by anything. There's only 1 atom per cell, so the molar mass of the element divided by Avogadro's number would give us the mass of that 1 atom. So the mass of the cell is equal to 4 times 63.55 divided by Avogadro's number. And please, again, make sure you're doing this on your calculator. You can pause the pencast and try it, so you're not just taking notes. Divided by 6.022 times 10 to the power of 23, and I get a very, very small mass, right? This is the mass of one little tiny unit cell made up of just 4 atoms of copper. So its mass, not surprisingly, is very small, 4.22, I guess 4.221 if we want to follow sig figs, times 10 to the minus 22 grams. Very, very small. We want the volume of the cell as well. Well, before I get the volume, I'm going to take the length of the side, which was 362 picometers, and I'm going to just take this chance right now while it's in a length unit, not in a cubic volume unit, and I'm going to switch it to centimeters because the question wanted the density as grams per cubic centimeter. So to switch to centimeters, I'm going to use 2 unit multipliers. The first one will get rid of picometer, and I'll use that recipe I gave you on the side of the page to switch picometers to meters. Okay, 1 picometer is a very small distance. It's 1 times 10 to the minus 12 meters. Now you could have switched that around. You could say 1 meter on top would have, if you put your hands out, 1 meter. If picometers are really tiny, there's going to be a huge number of picometers. So if you switch this unit multiplier around, you could have said 1 meter has 1 times 10 to the positive 12 picometers, right? 1 times 10 to the positive 12 would be a huge number of picometers. Since we're talking instead here of just 1 picometer, it's got to have a tiny, tiny distance, 1 times 10 to the minus 12 meters. Now we can get rid of the meter and switch to centimeters. We're on more comfortable ground now. 1 meter has 100, or 1 times 10 to the positive 2 centimeters. Okay, doing some mental math or grabbing your calculator if you want, that's going to give me 3.62 times 10 to the power minus 8 centimeters. So notice we switched the length of the side while the units were not cubed yet, they were just picometers. We switched to centimeters so that when we find our volume, it'll be in cubic centimeters. If you don't do this, you could find the volume and express it as cubic picometers. That's fine. And then use the same 2 unit multipliers to switch cubic picometer back to cubic centimeter. But you know how unit multipliers work. If you're going to switch a cubic unit, you're going to need to use each of those unit multipliers not once, but 3 times. So I think it's easier to do the conversion of units while it's still a length unit, picometers to centimeters, rather than doing it when it's a cubic unit, cubic picometers to cubic centimeters. So now we have the length of the side of our cube expressed in centimeters. We want to now find the volume of the cube. Well, this is just a cube, so the volume of the cell will equal S cubed, right? The length, the side times side times side, equals 3.62 times 10 to the minus 8 centimeters. Be sure you're putting your units in. Cubed. Grabbing your calculator, you can pause here and do the calculation. 3.62 times 10 to the minus 8. Be sure you're using your scientific notation button on the calculator. You have a little button on the side of the graphing calculator on the right side above the division symbol that looks like a little hat. That would be to the power of, or raised to the power of, 3 equals 4.74 times 10 to the minus 23 cubic centimeters. There's the volume of the cell. And again, this is a tiny, tiny little unit cell in the crystal lattice. Its volume has to be really small like that. Now we're ready to calculate the density of the cell. The density is the mass of the cell divided by the volume of the cell. The mass we'd already calculated was 4.22 times 10 to the minus 22 grams divided by the volume, 4.74 times 10 to the minus 23 cubic centimeters. Be sure your units are put in there. 4.22 times 10 to the minus 22 divided by 4.74 times 10 to the minus 23 gives me a density of 8.90 grams per cubic centimeter. Okay, so there we go. We've got the density of copper calculated, not measured, right? Grade 9 science, we measured these things in the lab. Here we've calculated it, knowing some information about the crystal structure and knowing something about the unit cell's length of a side. All right, question 10.87 tells us that tungsten, element W on your periodic table, has a body-centered cubic structure with an edge length of 317 picometers. It asks us to find the length. This is a bit of a strange question. What is the length in picometers of the diagonal of the unit cell? Okay, the diagonal passes, of course, through the center atom in the cell. Question 88 tells us to calculate the radius of the tungsten atom as well. So let's just do both of those. This should be an easier question. If you know what you're doing, you can pause here and just try it yourself. Okay, using the same approach that we did before, element tungsten, W, BCC structure. So we think about what we know. There are 2 atoms per cell, and we know that they touch along the diagonal, the atoms rather, touch along the diagonal of the cube. So this time the root of 3 times S is equal to 4R. We realize that we're not asked anything about density in this question, so we don't actually need to go get the molar mass of the tungsten. Okay, so don't automatically do things exactly the same way. The question is actually pretty easy if you stop and think about it. We want to know the diagonal of the unit cell. Well, if you remember where we got this formula from, either side of that formula, root 3s equals 4r, either side represents the diagonal of the cell, right? The atoms touched along the diagonal of the cell. That's why when 3 atoms touched along the diagonal, that's where we got 4r from, right? The 4 radii involved in that. So 4 times, if I know the radius of the atom, 4 times that is the diagonal of the cell. On the other hand, the diagonal of any cube is equal to root of 3 times the length of the side of the cube. So if I know the length of the edge, or the side of the cube, root 3 times that is the diagonal. So that's the root we're going to take. The diagonal of the cell is just root 3 times S is root 3 times 317 picometers is square root of 3 times 317 is 549 picometers. We also wanted to know the radius of the atom, also expressed in picometers. Well, looking at our formula, root 3s equals 4r, the radius of the atom would have to equal root 3s divided by 4. So root 3 times 317 divided by 4. Of course, we already knew that root 3 times 317 is 549, so we could have simply divided that by 4. When we get a radius of 137 picometers. If we wanted to go ahead and find the density of the tungsten, we would at this point. In fact, why don't we just quickly do that as a bit of an extension. We're going to need to look up the molar mass of tungsten, so we grab a periodic table. The molar mass is 183.84 grams per mole. Let's do the same approach that we did in the last question. We'll calculate the mass of the cell. This time it's 2 atoms per cell, so we'll say 2 times the molar mass, 183.84, so the mass of the cell, 2 times 183.84 divided by Avogadro's number, gives us a mass of 6.11 times 10 to the minus 22 grams. Now we'll find the volume of the cell. The volume of any cube is S cubed, but the side was in picometers, so let's go off to the side here and convert that. So the length of the side, the edge length, was 317 picometers. So we'll use 2 unit multipliers again, picometer to meter, meter to centimeter. So 1 picometer is a very small distance, 1 times 10 to the minus 12 meters. 1 meter is 100 centimeters, so 3.17 times 10 to the minus 8 centimeters. So let's take that and cube it. .17 times 10 to the minus 8 cubed is 3.19 times 10 to the minus 23 cubic centimeters. So now we have the mass, we have the volume, the density of the tungsten would equal its mass divided by the volume of the cell. So the mass was 6.11 times 10 to the minus 22 grams divided by the volume, which was 3.19 times 10 to the minus 23 cubic centimeters. 6.11 times 10 to the minus 22 divided by 3.19 times 10 to the minus 23 gives me a density of 19.2 grams per cubic centimeter. Be sure you put the units in all the way through your calculation so you can tell if you've forgotten to do something. Alright, let's do one last example together. This time we'll do number 10.89. It's basically a reverse of the question that we've done. We're told sodium has a density of 0.971 grams per cubic centimeter. We're told that it crystallizes in a body-centered cubic structure, which we know means there are 2 atoms per cell, and we know that in a BCC structure the root of 3 times S is equal to 4r. The atoms touch 3 of them along the diagonal of the cube. We were given a density, so we're probably going to need the molar mass. The molar mass of sodium from a periodic table is 22.99 grams per mole. We're asked to find the radius of the sodium atom, calculate the radius of sodium atoms, and the edge length of the cell. Okay? So, where to start? Well, we know the density. That's different this time. Before we were finding a density. We know a molar mass. Well, we know what we've done with that in the past, so why don't we just do that? We're not quite sure where we're going, but we know that at some point we can find the mass of the cell. It's going to equal, let's see, it's a BCC structure, so 2 atoms of sodium, so 2 times the molar mass, 22.99, divided by Avogadro's number. Not sure why we're doing this, but we can find the mass of the cell. Okay, so 2 times 22.99 divided by Avogadro's number. The cell has a mass of 7.64 times 10 to the minus 23 grams. Now, what could I do with that if I know the mass of the cell? Well, let's just go back up. We've used the molar mass. We've already used that. We've used the fact that there are 2 atoms per cell when we multiplied by 2 in the finding the mass of the cell. There's 2 things we haven't used yet. We haven't used the fact that root 3s equals 4r. Now, since we're trying to find the radius, we're probably going to use that last. So we're going to need to know s in order to find the radius. So how can we find s? Well, s is related to the volume of the cube. And now all of a sudden I see the answer. I have the mass of the cell and I have the density of the cell. So let's do a little sort of grade 9 calculation. We could use the formula D equals m over v, and we can rearrange it to find v. V will equal the mass divided by the density. In my experience, most students have trouble with that, okay? Or they try to memorize it. They don't think through the algebra, and half the time they'll say mass divided by density, half the time they'll say density over mass, and sometimes they'll even do some weird things with multiplying. So if we can avoid doing that formula in algebra, it would be better. Why don't we look at this as a unit multiplier? If we take the mass, 7.64 times 10 to the minus 23 grams, and we say to ourselves, I want to get rid of grams, and I'd like to know the volume, cubic centimeter. Then we recognize from those units that this unit multiplier is our density. The density was 0.971 grams per cubic centimeter, so for every 1 cubic centimeter. If we examine what we're about to do with the unit multiplier, we can see we're going to do the same thing as you would with the formula. We're taking the mass and we're dividing by the density. So 7.64 times 10 to the minus 23 divided by 0.971 gives me a volume. So the volume of the cell is 7.86 times 10 to the minus 23 cubic centimeters. Put those units in. If I know the volume of the cell, we could find the length of one side. We remember that V equals S cubed, which means the side is the cube root of the volume. Well, another way to say that is that it's the volume to the power of 1/3, which is how I prefer it using the graphing calculator. So S is equal to our volume, 7.86 times 10 to the minus 23 cubic centimeters to the power of 1/3. So on my calculator, 7.86 times 10 to the power of, using the scientific notation button, the EE button, negative 23, raised to the power of, that's that little hat button again, right above the division button, and then 1/3, I know that 1/3 as a decimal is 0.33333 repeating, so I'm just going to type in 0.333333, like several threes. Don't just type 1, don't just type 2, type 4 or 5, equals 4.28 times 10 to the minus 8 centimeters, right? Cubic centimeter, when you cube root, will give you centimeter. We're going to want the radius in picometers. I'm not sure it said that, but we'll express it in picometers. So now that we have the length of the side, we'll convert that to picometers. 4.28 times 10 to the minus 8 centimeter will go centimeters back to meters, and meters back to picometers. A meter has 100 centimeters. 1 picometer is a very tiny 1 times 10 to the minus 12 meters. So this is going to be 428 picometers. That's the length of the side of the cube. And now we're ready to finish. We wanted the radius of the atom, so using our formula above, the one formula we haven't used yet, the radius will equal the root of 3 times S divided by 4. The root of 3 times 428 picometers divided by 4. So the root of 3 times 428 divided by 4 is 185 picometers. There's the radius of the atom. We wanted to know the length of the edge of the cell. Well, we got that earlier. So there are the two answers that we were trying to find. So I hope that helps. There's some other questions on this worksheet that you could try that would be similar for practice. That would be number 85, 10.85, number 86, number 90, number 91 is a bit of a different problem. Let's see if you can try that. If you're done those questions and you had no problem, then you could also try working on some of your web